All right, today we are going to be doing part one of your DNA lab. And it's going to get a little hokey at the end because we aren't actually going to be able to sequence any of this DNA or do any PCR with it, but that is okay. I want you guys to see what the actual process of extracting DNA is like. So in order to do that, we're going to need a few different things. You're going to need a cup. You are going to need your test tubes, at least one of them. You're going to need the alcohol that I gave you in your kit. Some of you have a larger tube, and that's okay. You are going to need a measuring cup, and yeah, you're also going to need to put your alcohol on some ice to keep it nice and chilled. You are going to need some kind of measuring spoon. You're going to need dish detergent, and I don't know if I said this or not yet, but you're also going to need salt. So let's look at the procedure and see what it says here. So, procedure, make saltwater solution. How do we make the saltwater solution? All you need to do that is you are going to need, I think I wrote it down. Where did I write it down? You need a ½ teaspoon of salt into 1 cup of water. So here's my 1 cup. Here's my ½ teaspoon. If you don't have a teaspoon measure, you can certainly get by estimating using your scale, but that's going to be quite a bit harder. You're going to need about 3 grams of salt to make that. You're also going to need to make your salt solution. And to make your salt solution, you need 1 volume of soap to 3 volumes of water. And you only need a teaspoon of this. So what you could do is just take 1 teaspoon of soap into 3 teaspoons of water, and then just sort of mix that up and scoop out a teaspoon and put that into your separate cup. Okay, so what do we have thus far? We've got our salt water solution, we've got our soap solution, we've got our alcohol. We are all ready to go. So what you're going to start by doing is you're going to start by swishing a couple of teaspoons of that salt water solution we just made, so you can slurp it up and swish that for about 30 seconds. You want to time it, and you really want to get 30 seconds. You can go a little longer, but you want to get as much DNA as possible. I would also recommend washing your mouth out because if you're like me, you like to drink coffee, and it can get pretty gross looking if you don't swish your mouth out beforehand. I'd also recommend having like something sweet because salt water doesn't taste great. It's not the worst thing ever, but you can eat a piece of candy after you're done swishing the salt water. So we've got our solutions made. We're going to swish for 30 seconds, and then we are going to spit from where we were swishing directly into our cup of detergent. Okay, and then when we're done with that, you're going to want to gently move this around for 2 to 3 minutes. You don't want to shake it super hard. Just gently mix it around. If you do shake it too hard, you can actually cause the DNA to shear because it is quite a long molecule, so you can just break it in half. Okay, just mix that around for a couple of minutes, and then what you're going to want to do is get your measurement spoon, and you're going to take 1 teaspoon of the alcohol. Just get some alcohol on our spoon here, and you're going to want to just gently pour it on top of your DNA and soap mixture. Okay, it should sort of sit on the top. The reason you're doing that is that DNA is not soluble in alcohol, and so it's going to cause that DNA to get separated from the broken down cellular components that we created when we mixed it up with the salt. So the salt is helping to get the cells out of your mouth. The soap is helping to break those cells open and break open those nuclear membranes so that the DNA is just sitting in the solution. And then the DNA actually will not dissolve in the alcohol. And what that causes is this sort of mucousy like stuff that's just going to sit on the surface. Okay, so we've got all these processes done. We've swished, we've mixed that up for a couple of minutes. We've added alcohol to it, we let that sit. When you're done with that, you're just going to pour your solution into your Petri dish. Okay, just pour the whole solution into the Petri dish, and then you can use like a toothpick is probably the easiest way of separating your DNA, and ultimately you're going to get your DNA into a test tube that's got some ethanol in it. Okay, and there are points associated with actually getting your DNA into your test tube. So if you mess it up, you just kind of have to try it again. But yeah, you should get a little segment of DNA into your test tube. Then, like I said, we're going to get a little bit hokey, and we're going to pretend to PCR a certain segment of your DNA, and I'm going to talk about what's going on, what is PCR, why would we do that, and then we're going to pretend to sequence a little segment of your DNA as well.